Today I talked about the connection of integrin cell adhesion receptors and DNA repair. I think this is very novel, and we were the first to touch this subject, because we know that like when you inhibit beta-1 integrin, which is one of the central integrins, then you radiochemosensitize cancer cells. And so particularly when you use DNA damaging agents, then you obviously think it's as a next logical step that you address whether this also impacts on DNA repair. And so we investigated whether there's a mechanistic connection between the membrane signaling of beta-1 integrins and events in the cell nucleus with regard to the repair of DNA double-strand breaks. Well, we did that in head and neck cancers, because head and neck cancers desperately need optimized therapy. In the last 30 years, hardly anything had changed, and so we are looking for optimization of therapy by biological modulating receptors or protein kinases. And to be more physiological, we have established over the last 10 years 3D matrix-based cell cultures in which cells are embedded in a physiological matrix similar to the body. And this really impacts on all levels of cell physiology, like genetics, epigenetics, signal transduction. And so that is one of our models which we use in over 90% of our projects. And the other thing is that we use tumor xenografts in nude mice. And with these two, we measure different endpoints like survival and DNA repair. And then we use other techniques to address the mechanisms like Western blot, immunoprecipitation, mass spectrometry, phosphoproteome arrays, just to have a broader view on the signal transduction, which is either mediated or modulated when we have a therapeutic intervention. Oh yes, we have shown that in the last 10 years or so in many different other tumor entities like glioblastoma, lung cancers, pancreatic cancers. So particular cancers which are cancers of unmet need. So addressing really glioma, pancreatic cancer, and head and neck. And also we have done this in hematological tumors, because all our things, obviously, when we talk about cell adhesion and how this impacts on therapy resistance, is also true for leukemia, for lymphoma, and not only for solid tumors. So this is of particular interest and importance. The impact of cell adhesion on their resistance is different, a little bit depending on the genetic and epigenetic alterations seen in these cancers. Nonetheless, it depends on the either integrins or other proteins which are associated with these integrins. When we target them, how sensitive they are or how susceptible they are on top of radiochemotherapy. And what is one of our really big main interests is that we want to identify bypass signaling pathways which are induced by a particular molecular intervention. So, for example, when we use either integrin targeting or EGF receptor targeting, which is already in the clinic, then we do not hardly know anything about which pro-survival signaling pathways pop up when we have this intervention. And this is important because then we can easily identify the signaling hubs which are important, and we can inhibit them, and then we see an additional impact on or chemosensitization. So with regard to multi-targeting, with regard to precision cancer medicine. Yes, this is very important because all the things we're doing on top of radiotherapy or even radiochemotherapy, because these conventional therapies will still be in the clinic in the next decade or 2 decades. Nonetheless, we are all interested in more molecular therapies and more individualized therapies. However, I think these conventional therapies, we have to always consider them because they will be in the clinic for long time, and all the molecular drugs will only be on top of radiochemotherapy. And so this is important because also upon radiation and chemo, resistance mechanisms are acquired and induced, and so combining this would give us another level or another dimension of opportunities. The question is that acquired or intrinsic resistance? And I think most of them is intrinsic due to genetic and epigenetic alterations and the microenvironment, for example, hypoxia or growth factors and all this, what is in the microenvironment, which really impacts on the intrinsic sensitivity to therapy, including radiotherapy. The question is, what exactly is the cause of resistance to radiotherapy is unknown. There are quite a number of groups which repeatedly irradiate cells and see different mutation patterns, and I think this is in the beginning of research. So we will see in the next 5-10 years.